Yeah, we are going live. Yeah, we are live now. Good afternoon.
Good afternoon.
Welcome to the next webinar by AMR Declaration Trust, where antibiotic fail, bacteria eating viruses may prevail. The webinar will discuss the role of bacteriophages in treating drug-resistant infections. AMR Declaration Trust is a public charitable trust founded on the principles of Chennai Declaration document and initiative. Our primary objective is to stimulate discussion and action and explore Indian innovation and ideas to tackle the global challenge of AMR. AMR Declaration has minimal funds. We have received small donations from doctors, but this is not enough to conduct academic activities. We decided not to charge any registration fee to spread the message to a broader audience. We are grateful to the doctors who gave small donation to the trust and pharmaceutical companies such as Sanofi, Galangen, Converge Biotech, Alnishape, Fusion, Viatris who have provided us with small, unrestricted grants for the webinar. And we have requested a few other companies like Cipla and Pfizer we have requested grants, and so that we can conduct these webinars without registration fees. The first phage therapy was in 2017, a decade before Alexander Fleming discovered penicillin. And that was well before the invention of electron microscopy. So they were not able to even to see the bacteriophages. There were multiple successful observational studies since then. However, the West abandoned the phage therapy because wonderful antibiotics were available. However, Russians and other Eastern European countries continued with the phage therapy and they became masters. Now there's a renaissance of phage therapy in the West because we have returned to the pre-antibiotic era. Today, we have three eminent phage experts in our country. We have Dr. Sanjay Chibber, who is an emeritus professor in Punjab University. We have Professor Gopal Nath from Banaras Hindu University. And we have Mr. Pranav Johri, who is a founder of Vitalis Phage Therapy. Professor Sanjay, most members in the audience know very little about phage therapy. I'm a senior infectious disease consultant. My knowledge about phage is very, very little. Would you please explain to us in a very simple, plain language what these bacteriophages are?
Yeah. Bacteriophages, essentially, these are the viruses which attack the bacteria. And they inject their DNA or RNA into the cell. They take up the host cell machinery. They multiply. The cell then gets burst. And these phages are known as lytic bacteriophages. Of course, there are two kinds of phages, temperate bacteriophages and lytic. In phage therapy, we use lytic bacteriophages because we want to kill the bacterium which is invading and which is causing the disease. So lytic bacteriophages are preferred as far as phage therapy is concerned. These are the viruses of bacteria. They are either DNA or RNA, single-stranded, double-stranded. They are the ones which bring about the lysis.
So basically, these phage are viruses that infect and take over the control of the bacteria and they kill.
Yeah, but they are very specific to the bacterium. And it's not that a phage which is acting against one strain of E. coli will kill all the strains of E. coli. No, it is not. It is strain specific also, bacterium specific and strain specific also. So bacteriophages, therefore, we cannot use a single bacteriophage, and generally we make a cocktail of the bacteriophages when we want to use them as a therapeutic agents. So they are host specific bacteriophages, and there are many advantages, therefore, as far as the antibiotics are concerned, when we talk about the phages as therapeutic agents.
So the phages are very strain specific. They can kill only one specific strain of bacteria. For example, some of the phages, they can't kill all the E. coli. They can only kill certain strains of E. coli.
No,
and this is probably...
Sorry, they may be different phages for the other E. coli strains. And therefore, we make a cocktail so that we cover maximum number of the organisms within the E. coli.
Okay, so you make a cocktail of different types of phages against E. coli, so that it covers all the strains of E. coli. I have read somewhere. Yeah. I read somewhere, because I knew very little about phages, so I was reading about phages today and yesterday to have some basic understanding. This is what I read. The world has 10 to the power of 31 phages.
Exactly. Yes.
More numerous than bacteria.
Bacteria, yes.
And I also read that 40% of the marine bacteria are replaced every 24 hours because they are killed by phages. So phages are so powerful, so ubiquitous. And earlier, we're not talking about, when we talk about antimicrobial resistance, we talk about we live among bacteria. They are the super creatures of the God. It actually looks like the phages are even superior to the bacteria. Phages control the bacteria.
You're very right. You're very right. They are abundantly present in our atmosphere, in water, soil, surfaces, everywhere within the body also. In fact, a lot of people ask me, is it safe to have bacteriophages? So I say, yeah, we drink, we eat, everything goes in is bacteriophages also. So these are very safe as far as mammalian host is concerned, because they only attack the bacterial cell, not the mammalian cells.
So you're stressing a point, sir. So for the sake of the audience, because it's a very complex topic even for my team. So I'm trying to simplify it so that they can follow. You're all well-known global experts in the phage therapy. So I'm just trying to simplify the concepts that you give us. I'm just trying to simplify for the sake of the audience. We are living among phages. We inhale phage, we drink phage, we eat phage, and we're using the same similar phages for the treatment. So you consider it as very safe kind of modality of treatment.
Because they do not attack the mammalian cells.
They don't attack the familial cells.
They are specific to the bacteria. They are specific to the bacteria because they have the receptors on the bacterial cell. The phage has their spikes at the end of their structure. Through the spikes, the spike attaches to the specific receptor on the bacterium, and then it brings up the tube close to the head close to the cell and injects the DNA. So there are receptors. Basically, I want to make a point. There are receptors on the cell to which the phage will attach, and then only it will be able to inject the DNA or RNA into the cell. If you do not have a specific receptor, the phage will not attach to the particular bacterium. Therefore, when we use phages as therapeutic agents, the normal microflora is safe. Because one is they are strain specific. The second is they will only attach to the bacterium against which they are targeted, which means they have to have the receptors on that particular bacterium. Then only they will be able to enter into the cell.
The phages cannot attack human cells because we don't have receptors for the phages, and the phages can attack only specific strains of the bacteria. So they cannot just run around and kill all the bacteria like our antibiotics do. Our antibiotics do that.
Exactly.
The collateral damage of antibiotics.
Yeah. They do not disturb the normal microflora. For example, if I'm having something for my gut infection, my gut microflora will be maintained. Only the specific bacteria, for example, I'm targeting Vibrio cholerae. Vibrio cholerae will be killed, but the normal microflora like E. coli and the other bacteria which are there in the GIT tract will be retained.
So we give colistin, we give meropenem, and we give piperazole, tazobactam, and they literally kill every bacteria.
Everything,
And we call the collateral damage of antibiotic treatment. And these phages are not like that. They're very specific. They kill only whom they want to kill.
Yes, you're right.
The wonderful quality of the phages.
Yes. Another quality of phages vis-a-vis the antibiotics. Here, if you give one dose of the phage, and if the host is there, you don't have to give the second dose because the phage will keep on multiplying till the host is available. It will kill, say, a cell, release, kill the others, infect the other cells, kill them. So this way, the concentration keeps on increasing till the host is available. Whereas in the antibiotic case, we have to give repeatedly the doses to maintain the antibiotic concentration.
Oh, thank you, sir. So that means these phages infect and self-replicate in a virus, lyse the virus, bacteria in the bacteria, and lyse the bacteria, and then attack the next bacteria. So basically, these bacteriophages are self-replicating and propagating and keep on doing their job to a great extent. Unless our antibiotic, they do their work and their body eliminate, and we have to give the next dose of meropenem after 8 hours. And phages are not like that's a second superiority of phages over antibiotics. Sir, have you got Dr. Gopal Nath? Have Dr. Gopal Nath joined? Dr. Gopal Nath hasn't joined yet. Mr. Pranav?
Yeah.
Pranav, you are the founder of Vitalis Phage Therapy, and your company provides service to the patients. And how did you get involved in this phage therapy? None of my patients have ever undergone phage therapy, even though on a daily basis I treat dozens of patients with extensively drug-resistant infections. I deal with UTI, difficult infections. I never referred any of my patients to phage therapy. How did you get involved?
I got involved because of a very personal experience, because I myself suffered from a multi-drug resistant infection. And I suffered from chronic prostatitis, which was caused by multiple strains of Staphylococcus and Enterococcus, and it was multi-drug resistant. And after being put on multiple courses of oral and intravenous antibiotics, my doctor in 2016 finally gave up on being able to treat my infection. And he said that we can only put you on symptom management regimen of medicines for alleviating your pain and your constant fever because of the infection, the pain, the urinary symptoms. And he said that we can't treat it. You can basically just deal with the symptoms, and we have to, literally, I'm quoting my doctor when he said, you will live with this infection and die with this infection. So when I realized that at the age of 33, I'm suffering from a condition to which our modern medical science doesn't have really an answer to, I started looking at alternative ways of treating multi-drug resistant infections. And in one of the research that I was doing, I came across phage therapy. And as I learned more about phage therapy, and as Professor Sanjay sir has just mentioned the advantages of phage therapy, I realized that this could be an answer, or potentially an answer to the problem that I was going through, which was a multi-drug resistant infection. And as I researched more, I got to know the major practitioners of phage therapy in the world. And I reached out to the oldest institute of phage therapy. And I went there in the end of 2016 for my treatment. And I needed 3 courses of treatment to fully recover from my infection. And when I was able to overcome my infection, that is when I decided to start an initiative to make this treatment, to increase awareness about this treatment, and to make this treatment more easily available to people in India. Because I realized that I had to go through a lot of learning and research and obstacles to be able to access phage therapy. And if I could make it easier for people to access it, then a lot more people would be able to undertake the treatment. So we started this in 2018. So it's been 5 years, and we've had more than now 200 people in India who have been able to undertake this treatment for a variety of different conditions.
So Pranav, you were collaborating with the Georgian the university.
Yes, the Eliava Institute of Bacteriophages, Microbiology and Virology. They are the oldest phage institute in the world, and I think the most experienced practitioners of phage therapy in the world.
Yeah, that's where I had gone.
That's where I had gone on for my treatment.
The Eliava is keeping on coming everywhere. Such a reputed center. A few years ago, it was 5 years ago, I visited Peragov University in Moscow to give a talk on antimicrobial resistance, and I was talking on trials on antibiotic drug-resistant infections, and these Russians were speaking on randomized controlled trials on phage therapy. And then only I realized, they have developed, to a great extent, and we are still talking about antibiotics and pan-drug resistance. So how did the Russians and Eastern Europeans continue their phage trials when the West got antibiotics and stopped the trials? Why did Russians continue the trials on phages?
I think the reason is they were the pioneers at that time in phage therapy. So they continued with this, whereas the West found antibiotics a better source of treating the infection. So they they shifted towards the antibiotics rather than the phages. And moreover, the previous literature, if you look at the previous literature on phages, is more in Russian and the other languages rather than English. So that was also difficult at the time to know about the phages actually.
So the Russians were doing good work, but they were publishing in Russian language,
Yes.
which rest of the world did not understand.
Yeah.
Dr. Gopal Nath, we can see you.
Yes. I'm here.
Good afternoon, sir.
Thank you. Good afternoon, sir, for joining.
Thank you very much.
You know, Professor Sanjay and Pranav were discussing about the basics of phage therapy. And, and I've been just wondering, and because I've been reading basics of phage therapy, phages do not always kill the bacteria. Sometimes they get integrated into the genome of the bacteria, the lysogenic cycle. And how does that affect the bacteria? Does that help the bacteria, or is that harm the bacteria?
Actually, phages are helping the bacteria, and this is the natural process because both have to survive. So they are the commensal system or mutualistic approach is there. So the phages should be there and the bacteria should be there. That's why there is a limited growth of both the things. And because that is essentially needed for the whole life chain on the earth. That's why the lysogeny is very important, and sometimes lytic cycle is also important. This natural process we are simply exploiting for the killing of the bacteria which are causing harm to us. Otherwise, in the gut, they are all the time, all the bacteria which are in the gut, in our body, all of them are having the bacteriophages being excreted from our fecal matter and also from our urine. So recently we tested the number of urine from cow, buffalo, goat, and human being, and we have got variety of the spectrum of bacteriophages against different bacterial strains. So urine is not sterile. Urine is full of bacteriophages. All the body fluids are full of bacteriophages. So there is nothing unnatural. Only one thing we are doing is that we are having the higher number of the bacteriophages to kill the bacteria which are overgrowing or outgrowing than the norm. So that is the basis of the phage.
You made a very interesting statement, sir. No body fluid is sterile. You know, we believe CSF is sterile. And I read somewhere that the CSF have phage genome. If you do a PCR for the phages, you can find out phage genome in sterile, so-called sterile CSF. So it's not sterile anymore.
They say genome, but they are live viruses present there because they are nanoparticles.
Yes.
So
there are not just genome, there are phage particles are present in the CSF.
Yes, definitely. Not only genome.
Okay. So majority of our gut phages are in the lysogenic cycle, living along with the bacteria.
Both state, lysogeny and the lytic, both keeps on going. So there is switch over whenever whatever is the need and the stimulus is coming from outside because they have to maintain the equilibrium.
Oh, so they usually they get along with the bacteria very well, but if they need it, they go and kill.
Yes. You see, all the terrestrial system, aqua system, human body system, everywhere the bacteriophages are viruses, maybe eukaryotic cells. The viruses are always 10 times more than the eukaryotic cells because the bacteriophages has to kill them. So that we are getting the whatever are the metabolites and the things being synthesized by these prokaryotes, they are becoming available to the higher animals and plants. So that is the part of the food chain. So we are just simply exploiting that chain for our purpose. That's all. The beauty is that you are developing the antibiotic in 20 years that is coming to the market. But bacteriophages, every day you can have the new strain. If the bacteria develops resistance, you can have another new bacteriophages which are killing them because in nature this keeps on going.
So it's the bacteriophages control the homeostasis, controlling the number of the bacteria.
Yeah, definitely. In the soil, in the ocean, and on the any living being or maybe plants or the animals. Everywhere they have to maintain the equilibrium.
I read somewhere that even the beta-lactamase enzyme of the bacteria are given by the bacteriophages. Even the Shigella toxin, diphtheria toxin are all given to the bacteria by bacteriophages. Is that right, sir?
Majority of the toxins are mediated by bacteriophages. They are giving the survival advantage.
So they help in bacteria, but they kill the bacteria as well.
Yeah. Whenever needed, whatever is needed. So they do that.
And Dr. Sanjay, would you please explain us the kind of the work you do in Punjab University on bacteriophages?
See, my work on bacteriophages is something like more than 1.5 decades old now. I've been working with bacteriophages. Initially, I started looking for bacteriophages. I looked at their isolation, their characterization, and then we started studying them for treatment of the multiple drug-resistant bacteria in vitro conditions. And I am targeting 3 nosocomial pathogens, namely the Klebsiella pneumoniae, Pseudomonas aeruginosa, and Staphylococcus aureus. And all my observation till today, which have been made, these have been made in animal models. We have used animal models. The mouse is the model which we use. So we have a respiratory tract model, we have a wound infection model, we have a urinary tract model, and we target both the planktonic as well as the biofilm forms of the bacteria, because the bacteria exist in 2 forms, the planktonic and biofilm. So planktonic is easy to kill because they are they're available to the bacteriophages. But in a biofilm, the bacteria are not directly available to the bacteriophages, because biofilm is a structure which is sort of a mesh of the polysaccharide material in which the bacteria are encompassed, and the surface does not allow the phages to enter into the mesh and reach the bacterium. So for this, we have discovered that there are phages which produce an enzyme which is known as depolymerase enzyme. And this depolymerase enzyme essentially helps the bacterium to dissolve the biofilm matrix, and therefore they get the access to the bacteria. So the chronic infection these days are being attributed to the bacteria in the biofilm mode. And therefore now what we say is that phage therapy is not only for the planktonic cells, but also for the biofilm cells. So in this way, I can say we can target both the acute as well as the chronic infections. As far as the second thing is, earlier it was known that bacteria, these viruses or bacteriophages can target only the extracellular bacteria. But there are certain bacteria which are intracellular. People were of the view that the phage therapy will not work against the intracellular bacteria. But in my lab, we have proved, no, this is not there, because once the phage gets absorbed onto the surface of the bacteria, for example, Staphylococcus aureus, and then it is taken up by the macrophage. So this takes the phage along with the bacteria inside the mycobacterium and kill the whole of the progeny which is being multiplying within the macrophage. And so this way we can target both the extracellular as well as the intracellular bacteria. So this is, at present, we are making the biodegradable wound dressings, in which we are incorporating the bacteriophages, the phytochemicals, so that we don't have to replace the dressings every day, number one. Number two, for a better an improvement in the wound contraction, the resolution is faster. So we are now working on the wound dressings, both the biodegradable or non-biodegradable wound dressings. So this is essentially the area. Yeah, we are working also on the CRISPR-Cas. CRISPR-Cas in relation. CRISPR-Cas is a inborn immunity within the bacteria. This acts when the phage DNA or any other DNA from the external sources enter into the bacteria. And the bacteria then, like a Caesar, it works like a Caesar and it degrades the DNA. And we are working on this, and now how this drug resistance can be stopped by using the CRISPR-Cas mechanism. So these are the two areas on which my lab is working at the moment.
Professor Sanjay, you again stress one more advantage of the phages. Our antibiotics are mostly inactive against the biofilm extracellular matrix. They can't penetrate and kill the biofilm form of the bacteria. But our phages can get into the biofilm because of their license, the toxins. Is that mentioned?
So, Doctor, the depolymerase enzyme. In fact, I would
Okay.
like to quote you. I will quote one of my studies over here. We use this depolymerase enzyme along with gentamicin. The gentamicin was not effective against the infection.
Yeah.
But when we gave gentamicin along with the depolymerase enzyme, it was able to kill the bacteria. And this is an in vivo study in the mouse model. And we were able to cure the infection. So we are using an enzyme along with the antibiotic. Now, capsule is there in case of Klebsiella pneumoniae. There is a capsule which is present on the surface. So this depolymerase enzyme degrades the capsule, and that is how the gentamicin gets into the cell and brings about the killing. Otherwise, the gentamicin is not allowed to penetrate into the cell because of the outer covering, which is not allowing it to enter into the cell. So this way we have shown it's not only the bacteriophages, it also helps the antibiotics also.
Yeah, so the phages get into the biofilm. Phages get into the biofilm and also help the antibiotics to get into the biofilm.
Not the phages, the enzyme which is being produced by the phages, the depolymerase. It forms a channel into this biofilm. And through this channel, the antibiotic will reach the cells which are lying within the biofilm.
Oh, okay. Okay. That's very interesting. That's very interesting. Yes. Dr. Gopal Nath?
Yes, yes.
Would you please, sir, would you please explain the work you do, the animal, and coming to the human data will come later, the animal experimental work you do on phages?
As Professor Chibber said, we have worked a lot on the animal models, especially looking the effect of deciding the doses, optimum dose of the bacteriophages and the frequency of the doses. What is safe or unsafe? Because whenever there is the bacteriophage given in an inappropriate dose, it might be possible that they are killing the bacteria in a burst, and a lot of endotoxins are getting released, and this will further add to the shock because of endotoxin, that is endotoxemia, and patient will die because kidney and liver are unable to cope. So for this, we studied on the in the mice model against the Pseudomonas aeruginosa, Acinetobacter, Klebsiella pneumoniae. We are studying on the Enterococcus faecalis, all the escape organism, Enterobacter, and already we have worked on the Staph aureus osteomyelitis cases. And we found that in case of acute septicemia, when patients are admitted to the hospital, we have looked with the clinical perspective. One is that the patient is coming to the ICU with the infection, maybe a 6-hour-old, 12-hour-old, or 24-hour-old, as happens, or maybe the patient will develop the septicemia when admitted to the hospital. So in these conditions, what should be the dose? So what we found that if you are giving the higher dose at the earlier hours, maybe 6 hours before and after the setting of the infection, the mice are dying. But if you reduce the dose to the 10 power 5, then the death is not there. So we found that this 10 power 5 PFU per mice dose is safe. Number one. Number two is that if you are giving if the mice is being treated 12 hours or 24 hours later, you can give the higher doses as well, but they are not killing the mice. So it is safer that you are giving the small doses and you are monitoring the vitals of the patients, and you can give the frequency or different doses and number of doses according to the monitoring of the patient, which is appropriate and not leading to or not further adding to the septicemia. So patient will not die because of the septic shock, which happens usually in the clinical condition. Patient is admitted to the ICU. Doctors think that I should pour into all the types of antibiotics because they have to give it empirically. And they are usually antibiotics which are acting on the cell wall like penicillin and cephalosporins. So they are further killing are lysing the cell wall, leading to a release of the endotoxin. Bacteriophages are acting the same way. They are releasing a lot of endotoxin. So we have to manage the slow release of endotoxin by deciding the doses and frequency. number one.
Oh,
Number two, we did this study in the chronic osteomyelitis and acute osteomyelitis, where the usually in the clinical practice, it is the suggestion that you amputate. So to avoid the amputation, we did this study in 2016, it was published. And then we found that the bacteriophages are really able to cure the osteomyelitis. And then, because as Dr. Chibber said, they are able to penetrate the biofilm, and in chronic infections all the time, this biofilm formation is a natural process. And it is also very interesting that these biofilm are the niche or safe haven for the bacteriophages as well. So they are destined to live in the biofilm. That's why they are producing the polymerase enzyme. They are going inside, and they may go into the lysogeny, or they may go into the lytic cycle. If you are giving the higher number, MOI is higher, then the bacteria will be lysed. If the MOI is less, then they will go to the lysogeny. So that is in case of that one. So all the 5 bacteria we have taken in the mice model. Another model, Pranav is sitting here. On this case, we are just trying in the chronic urinary tract infection and acute urinary tract infection. And we decided the doses and the frequency or number of the doses for and we found that which route is safe. So recently we have concluded. One paper is already in coming in this month only. This is the this current microbiology. Today I completed this galley proof. Another paper is ready. That is on the Klebsiella pneumoniae chronic infection. That was on E. coli in mice model, and we found that about 7 to 8 doses per rectum or per urethra are safer than the oral route, than the peritoneal route, than the subcutaneous route or intramuscular route. So per rectal and per and that cures the infection. So that is these two things we have done in the animal model. Earlier we have already done the healing of the wounds in the mouse model by creating burn mouse model. We have done already, published in I think 14 or 15, something like this. So this is experience with our animal model. Another thing we are trying to find out, the lot of bacteria gut microbiota is associated with the metabolic disorders, neurological disorders, including the psychiatric disorders, and also autoimmune diseases. So many things are there. So we are trying to find out which bacteria is associated with this one. And we are just trying to remove that particular bacteria and looking the effect of the eradication of that particular bacteria from the gut, because they are very smart bomb-like things. Only they will be eradicating the targeted bacteria. So on the basis of Koch's postulate, we can establish that this bacteria might be associated with this particular disease, as we have already done or we have seen in case of rheumatic fever caused by streptopyogenes. So that philosophy is there. We are trying to find out the treatment for the obesity. We are also trying to find out the non-alcoholic fatty liver disease, which is the real basic problem. Nobody knows what is that, and autobrewery syndrome also. So maybe one day we are able to find out. We have created the Klebsiella pneumoniae model for the autobrewery syndrome and alcoholic fatty liver disease. Klebsiella pneumoniae has also been found associated with the obesity, and we have shown in the mice model that there is reduction of the weight, decrease in the weight by 30% when we are eradicating the same bacteria, Klebsiella pneumoniae, from the gut. So that has been published in the our journal published from the PGI Chandigarh. That is still has to be indexed, or that is not in the limelight, but because that is our journal, so that's why I completed. Another thing we have done on the Salmonella typhi, and because Salmonella typhi had been my basic subject for last 30 years, so we know that the chronic typhoid carrier causes the gallbladder cancer, which is very common in North India. I started from including Pakistan, Bangladesh, Punjab, Uttar Pradesh, and the West Bengal and Bihar. And this is associated with we have first time established that the chronic typhoid carrier state, chronic infection, which is found in the gallbladder, maybe in the liver, is associated with the gallbladder cancer. Then how to treat it? Because this is chronic infection. So in chronic infection, we created the mouse model. Recently, we got the 2 projects from one UPCST and another from the TST. And we have found that, as Dr. Chibber said, that the intracellular bacteria are also killed by the phages. We have shown that one. And also we are able to eradicate both the condition, acute typhoid infection and chronic typhoid infection as well. So if we can eradicate the chronic typhoid infection from the whole globe with the help of the bacteriophages, we can take care, or we will eradicate, as we have eradicated poliomyelitis, we can eradicate the typhoid fever as well. So this is my animal model studies. So if you are asking about
So,
the clinical,
yes, Dr. Gopal Nath. So I'll just summarize what your studies. Number one, you are working on the right dose of the phage, especially in early sepsis. Too little won't be effective. Too higher dose may be toxic because it will liberate all the toxins from the bacteria.
Yes.
And
Yes,
number
yes.
two, very interesting, the other interesting work you identify the right route of the administration and the right frequency of the administration. And much more than that, what we are really found, very interesting is we all know the role of microbiome on non-communicable diseases. The role of microbiome in brain disorders, in Parkinson, in Alzheimer's, in obesity, in diabetes. And of course, the bacteria are controlled by the phages. So you are working on the role of phages in these non-communicable diseases. And then you're working on, and we all know, you mentioned that the chronic carrier state can lead to including carcinoma of the gallbladder. So it will be, and also if we can eradicate the chronic carrier state, that will we can eliminate the typhoid just like we eliminate the polio. Wonderful, sir. Thanks. I'll come back to you now. Dr. Vasan Nagwekar is a senior infectious diseases consultant. He would like to ask a question. Vasan.
Yes, sir. Good evening, Dr. Gopal Nath. Yeah, good evening, Dr. Gopal Nath. I wanted to ask about what about cystic fibrosis where you have multi-drug resistant pseudomonas. It's almost impossible to eradicate that multi-drug resistant pseudomonas in cystic fibrosis because they are colonized with this bacteria. So, have any experience on bacteriophage therapy for this condition?
I think Professor Chibber will answer this question because he has worked on the respiratory model. And I think the bacteriophages do work on the cystic fibrosis very easily and it can be cured. Dr. Chibber, please.
Yeah, in fact, I have not worked on cystic fibrosis. I have worked on pneumonia basically, but I can tell you there are now reports in the literature, recent work which is coming up. It is very, very successful in cystic fibrosis patients also. Many centers are working on this and we have a published evidence now that this particular bacteriophage therapy works in cystic fibrosis patients as well. So this is a very good news as far as cystic fibrosis is concerned, because here phages can be given through the either in the form of powder or in the form of inhalers. They can straight go into the lungs and they can bring about the killing of the bacterium. So this is a very big success. In fact, I was reading 2 or 3 papers just yesterday only that it is very successful as well as cystic fibrosis is concerned. And I would also like to mention here, it is not only the bacteriophages, but it is the products of the bacteriophages. For example, endolysins. The endolysins are the proteins which are being produced by bacteriophages. These are also very, very successful. In fact, people are looking for their efficacy in the lung infections caused by Mycobacterium tuberculosis and the other related bacteria, because endolysins can be very helpful. These endolysins, they have certain advantages over the bacteriophages. Number one, is their specificity. Number two, it is their ability to lyse the bacteria very quickly. Number three, there is no problem of genetic material being transferred to the other bacterium, because this is one possibility which people talk about. And number four, it is not affecting the mammalian cells. So the endolysins are the ones which are now being worked into. In fact, there are many laboratories in China who are working on the endolysins, and they are modifying these endolysins within the laboratory to make it more effective. In fact, endolysins are active against both gram-positive and gram-negative bacteria. But at the moment, we have more success with the gram-positive bacteria, where they have 2 modes, the enzymatic mode and the cell binding mode, and therefore they are being very successful. So as far as respiratory infections are concerned, this is the strategy which is being worked into, the endolysins But cystic fibrosis, yes, it is very, very effective, Dr. Nagwekar. I would like to mention this for your information.
Thank you, Dr. Chibber. I wanted to ask one more question is, we have a lot of these chronic liver disease patients going in for transplant, and many of them are, their immune system is so bad that we are just not able to clear the bacteria. So will there in these chronic liver disease patients where the immune system is a problem, where even you have a sensitive organism, we are not able to clear. So will bacteriophage therapy work here, and we can have a collaboration to work on this as an experimental model?
I think Professor Gopal Nath is the right person to answer this particular query. Yeah.
Thank you. Thank you. Dr. Nagwekar, actually, you see, we have published 2 papers and we are looking for the immune response against the phages, which is a major objection by the NT people. And we gave the bacteriophages intramuscular and as well as IV and the intraperitoneal. And then we look for the neutralizing antibody development in the rat model, not in mice model. And we also gave a large dose of the bacteriophages published in Frontiers a few years ago, and just to see that what are the adverse effects. So we found that the already bacteriophages are there, naturally they are there, and we are simply using the higher number of the bacteriophages to kill the bacteria. And because they are the commensal flora, you know, when the immune system starts developing, the gut associated lymphoid organs do recognize the foreign antigens up to the age of 2 years. So by that time, the gut microbiota get colonized. So all these along with the bacteria and also along with the bacteriophages, respective bacteriophages. So these bacteriophages are possibly taken as the normal cells or the commensal cells of the own body, human body, and our immune system does not respond. That is why when we give in the large dose, usually for 3 weeks, there is no neutralizing antibody or whatever antibody we are detecting, they are not able to neutralize the viruses. So this is the reason. So it is very safe. And also we studied that when the neutralizing antibodies are developed, we can switch over to the viruses which are antigenically different from the previous cocktail. So the answer is there. The neutralizing antibody development or immune response is not a huge problem. And sometimes it helps in the immune modification also. So we should not bother about the development of neutralizing antibody. And we are still working on it. We are looking for the cell-mediated immune response as well.
Dr. Ashwini? Ashwini?
Yes, sir. So, sir, just now we have discussed about the wound dressing and role of phage therapy in that. So, we have lots of diabetic wound and bone and joint infection wherein prolonged use of antibiotic is required. So, do we have any model wherein we have tested it, whether it works? Because that will be a huge contribution to AMR. We could save long-term antibiotic in that way.
Yeah, ma'am, I would like to say that as far as the diabetic foot is concerned, this has been shown to be very successful. And in fact, at the moment, we are working at the burn wound dressings. And the MDR can be tackled very easily. And it has a rapid regeneration of the cells also, because the bacteriophages, they help the wound to regenerate as well, because you are not removing the bandage every day. So there is no mechanical defibrillation of the wound. So therefore, whatsoever is being formed, it is being stayed. So now both biodegradable and degradable wound dressings are being made. We are incorporating the bacteriophages, we are incorporating the phytochemicals and the other agents, where we are getting a lot of success in treating the burn wound infection in the mouse model only.
Can the same be applied to bone and joint infection?
Yes. It should, I think so.
Especially wherein there is an implant and it's difficult to take out the implant, so probably.
Yes, I have worked with implants in my lab. Yes, the K wires. We have worked with K wires in the orthopedic problem. So we have what we have done is, because majority of the time what happens is the wound gets infected when we insert the K wire. So what we have done is we have loaded the bacteriophages on the K wires, and then we have attached this. We have done in the mouse model. The studies, the bones were attached with those K wires, which are loaded with the bacteriophages, and there was no infection as such vis-a-vis the K wire being used as such. So with the implants, the K wires, we have done this kind of an experiment in vivo also in the mouse model, and the success was seen. So I think it should work with implants also.
Thank you.
Is it Dr. Vasan?
No, sir, I already asked my question.
Okay, fine. Pranav, now coming to the human studies, you mentioned that you have already treated around 200 patients. Now, how do you organize this treatment? Do you first test these patients' bacterial isolates against the phages? Because in microbiology, bacteriologists do susceptibility testing. Do you do such kind of susceptibility testing against the phages? And how do you identify the patients? What kind of treatment you provide, and what's the response?
So the entire process, I mean, as I mentioned, I was the first known patient from India who undertook phage therapy. And then we started this initiative to make it more accessible. So the first step was to make it, look at the regulatory framework, which allows treatment to be taken on phage therapy to be taken. So on compassionate treatment basis, phage therapy can be taken as individualized treatment. The next step was, as Professor Sanjay sir and Dr. Gopal sir mentioned, that it is extremely strain specific, and phage is extremely specific to the bacterial type and strain that it will work against. So sensitivity testing is a very, very important prerequisite to do before deciding on what phage cocktails have to be taken as part of the treatment, have to be given as part of the treatment. So in October 2020, we started with the diagnostic testing with phage susceptibility testing here in India. We tied up with a diagnostic laboratory here. The microbiologists from the lab were taken to the Aliava Institute for training, to undergo training on phage susceptibility testing. And since October, more than 350 samples have undergone testing for phage susceptibility testing. The phage susceptibility testing is done against what are called the standard phage cocktails, which are, as Professor Sanjay sir mentioned, that these are cocktails which are produced, which are targeting the typical common pathogens. So that is the first level of testing which is done, where the phage susceptibility testing is done, and whichever cocktails are working, that is mentioned as sensitive in the report, and the ones which are not active are obviously either not applicable if they don't have phages against that particular bacterium, or if they have phages and they are not sensitive to the strain, then they are resistant. In case we come across a bacterial strain which is resistant to all of the standard phage cocktails, in such a case, a customized phage cocktail is prepared, which is targeting that particular strain. So the treatment so far have all involved the local doctor being a part of the treatment. And the prescription, guidelines, dosage, all those details come from the treating doctor at the Aliava Institute. And then the local doctor administers the treatment to the patient following those guidelines and the prescriptions. In some cases where, the patient cannot even wait for the case is such an acute case where the patient cannot wait for treatment for even the testing to happen. And typically the phage sensitivity testing turnaround time is approximately 72 hours to 96 hours. So in such cases where even that time frame is not available, then we will often refer the patients to Dr. Gopal Nath sir, since he has a phage therapy unit and he has treated many patients with variety of different conditions. And that is typically how we are taking the prerequisite work for all the treatment before the patient starts the treatment under the care of the local doctor.
Yeah, Pranav, now I have a patient with recurrent urinary tract infection. All the oral antibiotics, including fosfomycin, are resistant. He has got prostatitis. And I don't have any option. I can't keep on giving colistin multiple courses to somebody coming to my OP. And if I want to refer that patient for phage therapy, initially, should I do a urine culture? And should I send you the bacterial isolates for phage susceptibility testing?
Yes, so we do have cases where bacterial isolates are sent to the lab here for phage susceptibility testing. In cases where the patient is unable to undertake the travel to come to Delhi, In such cases, the isolates are picked up by the lab's courier, brought to Delhi. The regular culture and antibiotic sensitivity is done. And once the isolate is identified, on that, the phage susceptibility testing is done.
And that will take around 72 hours.
72 to 96 hours, typically.
And then how will you administer these phages for this patient with the prostatitis?
So the administration modality differs from based on the condition. So as Dr. Gopal Nath sir mentioned that for urinary tract infections, it is given rectally and via urethral instillations or bladder instillations. For cystic fibrosis or respiratory infections, as Professor Sanjay sir mentioned, it is given through a nebulizer. So the modality differs. For wound infections, it will be typically done through a gauze dressing or a bandage dressing with the periodicity decided by the doctor. So it will the way it is given, the modality will differ based on the condition. But the idea is to make it as localized a treatment as possible. So for urogenital infections, it would be given rectally and via the urethra. For respiratory infections, it would be given through the nebulizer. For wound infections, it would be directly administered to the wound. For GI tract conditions, it would be through oral plus rectal suppositories. Those would be the administration modality.
So what kind of conditions are you treating at your center?
First of all, we are not a center. Basically, we are a facility. We are creating the framework so that people can undertake the treatment. The treatment is done by the local doctor. So, for example, if you mentioned if you have a chronic UTI patient, the guidelines and the prescription dosage all will come, say, from the urologist at Eliava Institute, but you will be administering following those guidelines to the patient under your care. We will create the bridge and the framework for that treatment to be enabled. So that is the framework that we are following. In terms of the process, what would typically happen is that you would either send the isolate to the lab in Delhi for phage susceptibility testing, or if your patient can travel, then the patient would travel, give the samples in person at the lab. The antibiotic sensitivity and culture and antibiotic sensitivity would be done and followed then by the phage susceptibility testing. Those reports would then be studied along with the relevant case history by the doctor at the institute. Then they would have a consultation with you as the local treating doctor and would propose a treatment plan. They would take your insights back into modifying the treatment plan if required, and then the phage treatment would be shipped to you, and then you would administer following the treatment plan.
Okay.
Sorry,
how expensive it is? How expensive is for one phage treatment? For example, a recurrent urinary tract infection.
So a full course of treatment lasts for about 10 to 12 weeks. So it's about 2.5 to 3 months, the full course of treatment. And it costs approximately 1.5 lakh rupees.
Okay, but how many times you have to administer in this 3-month period?
So typically the dosing period will go for about 15 to 20 days, followed by a break period of about 10 days. The break periods are built into the treatment because as Dr. Gopal Nath sir had mentioned about the endotoxin release, so it is built into the treatment to allow the body to detoxify from the toxins. I mean, the phages the concentration of phages used are typically 10 raised to the power 5 PFU per ml. But the break periods are built into the treatment to allow the body to detoxify from the release of the endotoxins.
Okay, interesting. Very interesting. Thanks, Pranav. Dr. Gopal Nath would like to add?
Sorry. I would like to add a point over here.
Dr. Sanjay, yes.
It is in such cases, it's not only the phages. They can be given in conjunction with the antibiotics also. As I've said, sometimes they act in synergistic mode. So it is not only the phages will be used for the treatment. Along with this, the antibiotic can also be given while treating a clinical case because both of them can work in synergistic fashion and can bring about the relief much faster. So this is also there in the literature.
So a combination of phages and antibiotic.
Yes, that's a very good point, sir, because in acute cases, like we have had a few cases of patients who are hospitalized and it has been a hospital-acquired infection. In such cases, these combination treatments have been done.
Given.
Dr. Gopal Nath, would I like to add?
Yeah, we are running the 4th clinical trial on the wound healing. 3 were on the chronic wounds, as Ashwini asked about that one. We have got we have published the papers. Even the 6-year, 8-year, 12-year-old chronically infected wounds are very easily healed by the bacteriophage therapy. It takes about 60 days to 90 days, and the total application is usually say 7 to 12 applications on alternative days. So it is very simple. And also we have tested on there was another clinical trial registered in CTRI. We just tested the bacteriophages in the acute this road traffic accident cases admitted to our trauma center, and we found that the healing process are the SSIs. This skin grafting can be done on 21 days if you are using the bacteriophage therapy, and if you are going to the conventional antibiotic debridement and antibiotic therapy, then it takes usually 42 days, and the success rate is much better. There is no scarring, and these problems are not there, and healing is quite fast. So bacteriophage therapy is good at acute wounds also, as well as in the chronic wound, even associated with the diabetic cases. So I think there are 3 publications on the chronic wound and 1 on the acute one. So this is another one we are running still on the chronic wound infections as a double-blind study.
So you're Dr. Gopal, you're doing the 4th clinical study on wound infections.
Yeah.
Okay, and you are getting an excellent result from phage therapy.
Yeah. Yes.
And also you just mentioned that in wound, if an antibiotic and the time for the wound to be ready for grafting is around 41 days, and with phage is around 21 days. So almost half.
Yeah.
The time is almost
Yes.
good success.
And the cost is 1/3 of the conventional therapy. That is
yeah, and antibiotics are too expensive compared to the antibiotic course, which runs into lakhs in severe infections.
Yes,
phage therapies look like far cheaper than expensive antibiotics.
Yeah.
And are you doing clinical trial on any other conditions?
No, not now. Actually, for the septicemia or injectable one, there is no permission. So that can be done on compassionate ground. If the 2 physicians are agreeing in case of the this septicemia, then we can go. But the clinician has to come forward. And we are planning to go for the pediatrics septicemia model. One of our professor is ready for that one. So we'll be submitting the project to the clinical this ethics committee. If it approves, then we can go for it.
You mean injecting into the blood?
Yeah.
So what is the problem in getting as I mentioned, the Russians are talking about randomized control trial of injecting the blood. Why are we not getting the ethics committee approval? What is preventing our authorities to give you permission to do a trial?
Authority says that we cannot inject the live viruses to the human.
Russians are doing it. Georgians are doing it.
They are doing, but actually we are trying to impress upon the regulatory bodies by doing all these works and research. So maybe in coming 5, 10, 20 years, are the people, if they are telling that this can be given, and you see, this is the natural process. We are simply exploiting this. There is nothing we are taking from outside. All your urine is passing the bacteriophages against Enterococcus faecalis, Staph aureus, E. coli, Klebsiella pneumoniae, Acinetobacter. Then we are simply increasing the number. I don't know when they will be agreeing and allowing us to use this.
Because in clinical practice, we treat patients where not even a single antibiotic is effective. And that situation is going to get worse. The most powerful antibiotic, the colistin. I treated plenty of colistin-resistant infections. I'm treating the most recent ceftazidime avibactam resistance, and the next antibiotic for the pan-drug-resistant Klebsiella that in South Asia will not be available even for the next 3 to 5 years. And we and the phage and the global literature, there are a lot of papers coming out of use of phage in sepsis patients, and we need to explore such methodologies because we need options. We don't have antibiotics, and we need options.
Yeah, in fact,
we need options. Yes,
I would just like to add that now in today's times, it is actually not only the Georgians and the Russians which are now doing it, because Belgium enacted the Magistral Phage Act in 2018. So they created the framework for phage therapy administration. Australia enacted a legislation last year called the SPARC, legislation, which also created the regulatory framework for phage therapy. So there are many countries which are different from Eastern European region, which have come forward and created the frameworks. So hopefully we should see a quicker acceleration of this even in our.
So Pranav, you mentioned we don't have a regulatory framework for phage therapy. We don't have a committee.
So currently it is only permissible as compassionate phage therapy, CPT, compassionate phage therapy on offer as individual treatment basis.
So we don't have, our regulatory bodies are not currently looking into this as randomized control trial. There's no no regulatory body. So who is responsible for it?
So, sorry, yes,
Dr. Gopal.
please go ahead. Yes, please go ahead.
They are asking that the bacteriophages should be fully characterized. Whole genome sequencing must be done. They should not have any antibiotic resistance gene. They must not have any virulence gene in them. And then their safety, this phenomena should also be checked thoroughly, and they should not excite the immune response as well. So that's why we are working on that. And then what should be the optimum dose for different conditions. So we are already doing that work so that the things are optimized on the animal model. And then we can jump into this one when the regulatory bodies are permitting. And we have to provide the enough data so that they are convinced that they are safe and they can be used. So still we are working on that.
So still waiting for all the conditions to be satisfied.
Yeah.
So our regulatory authorities ask for certain conditions, and our scientists are working to satisfy those conditions. And the conditions are satisfied, then of course regulatory authorities will look into it. So of course we are happy that the system in place and our regulatory authorities are sincerely looking into it. We all know that they are very sincere, because our regulatory authorities are very much aware of the antimicrobial resistance problem, and we know that they are doing everything to do whatever possibility. And we all know that our regulatory authorities and our government are encouraging the Indian innovation to tackle the global challenge of AMR.
In fact, this is the need of the day, because many times we get the call from people where the person is on the deathbed and they want to try phage therapy because they beg from you that you give us the phages so that at least our patient can be saved. So this is the need of the hour, and we must go ahead with all this. And in fact, in India, we have this SBRT, Society for Bacteriophage Research and Therapy, where we are encouraging the young scientists to come up and work in this direction. So that is the basic aim of that society. Dr. Gopal Nath has formed that society long back.
Can I add something?
Yeah.
Oh, Dr. Rama, yes, I was planning to come to you, please.
Yeah. Thank you. Actually, I'm traveling, so I will not be coming on the video. But
your voice is breaking, Dr. Rama.
Because can you hear me?
Yes, now, yes.
Yeah. So what I'm saying is that it is very important to characterize the bacteriophages. So, our endeavor has been towards that. So some of the phages we have isolated, we have done total genome sequencing exactly to see that what are the genes present in that. And we have to see that the lytic gene should be there, and integrase and other holofin genes and all other genes are not there. So these sequences we have submitted to NCBI. And once the total characterization is done, then you are 100% sure, because the clinical trials as of now are not possible. And even animal studies are a lot of restrictions there. I think Dr. Gopal Nath is lucky he is able to do it. But it's not that easy nowadays because of animal ethics. So the fully characterization of the bacteriophages is the key. And we have already published some of the phages, and we have deposited these sequences in the gene bank. And I'm happy to report that we have isolated some novel phages, which we have reported in Microbe Resource, a journal of American Society for Microbiology. So this particular phage we have isolated against a rare strain of Pseudomonas, which causes human infections also. And perhaps this is the first phage in the world which we deposited. So I think these things are, expensive. And unless ICMR and other funding agency comes forward with a lot of funds to support these kind of work, then only you can have, the characterization, the clinical trials, and the need is to have our in-house phage repository. Yeah. And there is one institute in Hisar, which is keeping some of these phages, and they are depositing the phages there. And also one ICMR center has started doing it. But even now, there are only isolated studies and isolated phage repository. So, we really have to join hands. And I'm happy Dr. Gopal Nath and Pranav, they've all been working together. We are part of the common project, especially with Dr. Gopal Nath. And it has been possible to mutually train the students under those people who are already expert. Our students went to Dr. Gopal Nath in the beginning, and now they are working on their own. And also we could train, AIMS could train some of the students in different laboratories to do the phage isolation. So everything is required. You need training, you need awareness, and you need a total characterization of the phages so that we can get that right kind of approval to go ahead with that. And I think the kind of webinar you have organized is commendable, because this is required to create awareness. So unless you have awareness, unless words come from people like you, it's not, everyone doesn't believe easily. And I think these kind of webinars will definitely.
Ma'am, you lost your voice.
Interest.
Yes.
I said it will instill interest in the bacteriophage research. And I personally talked to the ICMR director, Dr. Bahel, the present director, and he is definitely moving towards that direction of giving funds and especially recognizing bacteriophage as a priority area. So this is all I wanted to add. Thank you.
Thank you, Dr. Rama, for your very valuable comments and suggestions. So, and we all know that, we need to discuss, because we don't have, as clinicians, we don't have treatment options. And we, as you mentioned, Dr. Rajiv Bal, we all proud of him as our DG ICMR. And we are happy to know that Dr. Bal is showing a real interest in phage therapy and supporting the scientists. Thanks a lot, ma'am. Dr. Vasant, you would like to make a comment?
Yes, sir. Sorry, I'm not keeping video on because I'm traveling. So I just wanted to ask, I did hear about everything, that it's very difficult to get the phage therapy. But suppose if we require for some difficult patient, do we require to take a DPG approval and all that to go ahead, or we just can't be at this moment with phage therapy?
Pranav, would you please answer that question? Dr. Nagwekar asked us.
I couldn't really hear.
What Dr. Nagwekar asked is, suppose if one of his patient needs phage therapy. Okay, to get that, do you need DCG approval, or is there any easy way? Or can we use it on a compassionate basis? And which condition we can use on a compassionate basis, and which condition we cannot use compassionate basis and needs DCG approval? Dr. Rama, we can't hear you.
No, I just said that DCGI, I have tried to contact. They want all the details. And as I told you, that characterization of the bacteriophages is very, very important. So those details needs to be submitted to them before we can use our in-house bacteriophages. But as far as the usage of phages cocktail coming from Ilyava Institute, where they are already using it in human infections, that Vitalis Phage Center has already worked on, and on compassionate basis, you can send the strain, get the sensitivity done, or even in Delhi with Dr. Dang, and then you can, kind of get that right cocktail for the treatment. So you have to see if your patient is really sick, and if it is MDR strain, on compassionate basis with the permission from the patient family, these things can be worked out. But as far as in-house phages are concerned, we still have, some challenges to take care. So, in fact, I am also written to the DCGI for the approvals and all that. But some animal studies are required before it is done.
Okay, Dr. Rama, thank you. I will just summarize what Dr. Rama and Pranav told, so we can ask them more clarification. What I understand from the discussion is that if you want to use phages for the clinical treatment, we need evidence for the characterization for any sender. But for Eliava already is a well-known center. They have characterized the phages, and we can use their phages, but except for bacteremic, we can't inject into the blood in the India for the time being, but we can use it for other indications of phages like from Eliava or phages like by BHU for limited indications. Am I right, Dr. Pranav? Pranav and Dr. Gopal Nath?
Yeah, you are right.
Yes,
we can use
that's
the compensated ground. And
Yes,
because you have got no option, patient is on deathbed, we are going to lose that patient. There is no option left. Then in that case, two physicians, if you are there, they are agreeing. And then even uncharacterized phages can be given, because it is better to do something than die and letting the patient to die.
Yeah.
So
But we can't inject into the blood. We can't inject into the blood.
That will be injected in the blood.
Oh, it will be injected into the blood.
Yeah,
Okay.
Yeah.
Oh, okay.
But the issue is, in this case, the issue is that already the patient has got compromised the vital organs. This is quite late. So when we start, so it is very difficult to take out those patients. If you are doing in the beginning, like in prophylactic manner or in the earlier hours of the septicemia development, then it will work. So usually it is not effective if already the patient has gone to the non-reversible condition. That is the situation. So we have to take care of that one also. One can't say that the phages are not working. You know that the septicemia becomes the irreversible at certain stage when the vital organs like kidney and liver is not working. So in that case, we can't do anything.
But it will take 72 hours for you to prepare the phage.
No, actually, we have got the phage collections, and then we are having some certain broad spectrum phages collection, and we are having certain cocktails which can act on majority of the bacteria which are available. So if you have
ready-made phage cocktails.
Yeah.
Vasant, does that do this answer your question? Any more clarification necessary?
Yes, sir. Thanks so much. I just want to ask one more question. What is the major disadvantage or adverse events which the relatives will be asking us when you're giving the phage therapy? Even in a critically ill patient, because always patients always ask, what is the side effect of this? We know they are not bothered about a multi-organ failure, but they always question what will be the side effect?
Dr. Vasant, actually, you keep just a bit away from the mic, so it is echoing.
I will repeat the question. I just got it. What Vasant asked was, suppose if we inject phage into the blood for a patient with sepsis and no antibiotic treatment options are available. The family is asking what will be the side effects of the phage? How can we talk to the family?
Actually, there is no side effect provided that we are giving the optimum dose and optimum frequency, and we are monitoring all the time as we do conventionally the vitals of the patient. So we are not letting the endotoxemia going very high. So you can give in drops, and you are just monitoring the oxygen concentration, saturation, and all those pulse rate and everything. So in that case, you know that when you have to stop. So you can give the small doses and gradual doses continuously. This is possible with the human being, which is not possible with the mice model. So this is the advantage in the human cases. So it can be done easily. It's not a problem.
Vasant, is that clear?
Yes, sir, that's clear. Yes.
Okay.
Yes,
now,
clear.
Yeah. Now, Doctor, my question is to all the three of you. So we are clinicians, we always ask for what is the evidence from the randomized controlled trial. So I did a Google search this morning, and I got a systematic review published this year where they looked into 2,000 published papers in English language or translated papers where full translation is available from other language, and they could identify 2,000 trials or large studies on phage therapy. Out of that, only 16 had done high quality clinical trials. Of that 16, 3 were before 2000, and they called it historic trials. Only 13 were post 2000, and they called the modern trial. Of the 13, only 7 looked at efficacy, and out of the 7, only 2 could prove superior to antibiotic, and 1 were in chronic ear infection, the auricular infection, and by Pseudomonas, and the other one recurrent rhinosinusitis because of Staphylococcus, where the phage was superior to the antibiotics. Where rest 5 were looked into efficacy, the phage therapy was not superior. They were not inferior, but they were not superior. But all the studies confirm the efficacy. Now, out of 2,000 studies, only 16 of them are high quality, and out of 7 for efficacy, only 2 could provide superiority. But the rest 5, the conclusion of the systematic review was, of course, phenophages kill bacteria, but it's very difficult to do randomized control trials on phages because of the specific microbiological characteristics and patient characteristics. Why is it so difficult to do clinical studies on phages?
Should I answer?
Yes.
Actually, you see, when we are conducting one first randomized control trial, double blind, but it becomes very difficult to take the consent from the patient. And then what we are doing is we are doing the conventional therapy to both the cases. And then and then they say that why the patient will be divide up that fast therapy, which is more effective, But what is happening, it is very difficult to make it double blind. So we know that the cases which are coming to us already, if they have been treated for 2-3 years, they have been failed. That's why they are coming to us. So usually these cases are there. So it is very difficult to name them that we are doing the double blind. We know that that is the real case control study like things, but double blinding is really very difficult in these cases.
And in clinical, even in clinical trials, you will be utilizing the cocktails, pre-prepared cocktails, and not personalized bespoke combination.
No. We are doing personalized as well, because when we are giving, we are isolating the bacteria, preparing the phages, and then we are giving. One group is getting the placebo, and another group is getting the cocktail, which is already personalized.
Because this conclusion of the systemic, of the systemic review was, yes, out of those 7 trials, only 2 could provide superiority, but rest 5 doesn't mean the phages will be working, but difficult to conduct the trial. The reason they mentioned was in individual case series, for example, Pranav will caught out of 200, majority would have been excellent. That because they are individualized, bespoke, prepared for that particular patient, and that works well. But in a clinical trial, it may not be possible for every patient to prepare the exact phage combination for that particular patient. There's one of the explanations given for the lack of superiority in clinical trials, but excellent performance in case series.
So what you are saying is very true. It is actually a personalized treatment, because the series, which the case husband case, so there also it has to be personalized treatment, then only it is effective. And yes, it is a challenge, but wherever the cocktails are available, we can give that. And if that cocktail is not effective, then we can look for the specific bacteriophage from our whatever phage repository we have. We can even isolate new phage. Only thing is that not many centers in India are doing it or are trained to do it. So that is, though it is not a very difficult technique, but somehow it is not very popular or not given so much of importance. So I think it is doable. Even personalized treatment can be given, but then we have to really create awareness and give boost to the research in bacteriophage area, which is lacking. So that is why, because most of the time the research is into, we are doing so much of research for MDR, but what we are doing to combat MDR, that is lacking. So we are spending crores of money to know what are the MDR pathogens, but we are not spending enough money to do the bacteriophage research so that we can come out with the answers. So I think that is what is required. And I must tell you, it is not just Ilyava Institute in USA. One young girl, PhD student in Boston, company. Just 3 of them, they started their company on bacteriophages, and now they're doing very good work. So a lot of entrepreneurship is required. I'm sure Pranav is very good example for that in India. and not even with science background, he has done such a wonderful job. So I think that kind of awareness is required. And if we have that kind of awareness, and we all must join hands, which is very important, then only we will be successful. So I think our voice should reach to the higher authorities to make it, accessible therapy to all the people in India who are having MDR infections. Or sometimes even, our former director was very, very positive towards that. And he said that why wait that patient becomes critical and then we will give.
Yes.
Dr. So, yes, sir, please go ahead.
Yeah, sure. No, you can go.
I was just
critical and serious.
Yeah, ma'am. Yes. We understand your question.
Yes.
We definitely need you scientists. You are doing Russians or Georgians, maybe the leaders, but people like you, you're wonderful scientists, dedicated scientists working for the country. You need support. Scientists like you, must get the support from funding bodies to do excellent research on bacteriophages. So our country can be, can compete with the Russians or Georgians. We can provide solution to the world, and you scientists need support from the funding bodies. There's no doubt about it. And Pranav, an example of entrepreneurship in bacteriophages. We need more entrepreneurs to come up in the bacteriophage field. Dr. Sanjay?
Yeah, I just wanted to share that whatever data you have presented, it is from the previous years. But now I find that there are many biotechnological companies which have come up in the recent years, and they are doing the clinical trials. The phase 1, phase 2, their success has been achieved. Phase 3, they are reaching. And there are many Elevia biopreparations, Locus Biosciences, Intralytics, Phage. There are so many companies which have come up in recent years. And this phage therapy market, in fact, is growing up in the advanced countries also. And sure, we will have an answer for all this. The success with the clinical trials will be there as far as these biotechnological companies are concerned, and they are into this particular area.
So, Dr. Sanjay, there are more entrepreneurs in India in phage technology.
Yes.
I also want to add something here. Sorry, can I?
Yes, Dr. Rama, please go ahead.
So Dr. Sanjay brought out very important point in the beginning. He said that not just the phages, even the phage products like endolysin. So far the studies have been more towards endolysin, which is for the Staph aureus. But we have got a project from ICMR, which we have started working, where we are trying to clone the endolysin gene against Acinetobacter. There are not many gram-negative endolysins available. So I think even those endolysin products, if we can have, or any cloned, proteins we can have, we will really be able to, kind of save so many questions and queries which are raised. All the side effects can go. The production is easier, and we can have maybe an endolysin which is effective against many organisms rather than just one strain. So these kinds of researches are required, and I think I'm really happy that ICMR actually identified that as one of the study to be funded last year, and good results are coming into that. So I'm sure even the phage products can be, something which can be looked into, and they can be used as therapeutic agents. This will be much more doable. Thank you.
Thank you, Dr. Rama.
I would like to add over here. The only problem with endolysin is their low bioavailability, as well as the low half-life is very short. And therefore, what people are doing, in fact, we have done in our lab, nanoparticles are being made, and the endolysins are being put into the nanoparticles, and they're delivered through the nanoparticles to increase their bioavailability, as well as to increase their half-life, so that they are more effective in vivo. Yes.
Yes, fully agree. Right. Yes. You are also right. I agree with you, because that is the ultimate purpose. Once we have the clone protein, then that is the next step. Yes. Thank you, Dr. Chibber.
Thank you, Dr. Sanjay and Dr. Rama. We have Dr. Mrs. M. and Sumana who want to ask a question. Sumana?
Yes, sir. Dr. Sumana here. I'm from JSS Medical College and Hospital. I'm professor of microbiology. I have a few students of mine who are doing PhD. Is it possible for me to send any of my PhD students to any centers they can train her and we do the susceptibility, or we collect all the MDR strains and do the bacteriophage susceptibility and do some kind of a work like that, is what I wanted to know.
It's better that you are sending your student for a few days, maybe 15 days or 1 month for training. That can be done. Otherwise, we have planned to have the workshop in June or July, and we'll be taking around 20 students for this purpose. So she can wait, or they can wait for that time, or they can come anytime to the lab, because all the time we are working continuously on these.
Yes.
That's a nice opinion. That nice to know that, because we will be distributing this, recording this webinar among medical community, among scientific community, among policy makers as well. And I'm sure Dr. Gopal Nath, you will get plenty of interest from so many candidates from around the country.
Sure. I know there will be overwhelming crowd. Actually, we have to pick up first come first serve like basis. We know that. So
I think
we'll be fixing at least 2 seats for our SSS.
I would also like to add that guidance can be given on phone itself, because it is not very difficult to isolate these phages. The literature is available, so they can seek guidance from us online also, if need be.
Any other questions from the audience? Dr. Nagwekar, you raise your hands.
Yes, I wanted to ask you, sir, anything to do with artificial intelligence, and will that be beneficial for bacteriophage therapy? And second question is this similar to a precision ID? Just like we have precision oncology, whether this is similar to precision ID? One is artificial intelligence and second is precision ID.
Yeah, basically, Dr. Nagwekar's question is, can we use the artificial intelligence to, fine-tune the phage therapy?
Yes. Can I answer this?
Yes, ma'am.
I think artificial intelligence nowadays is being used in many areas, and it's a very good suggestion, or I must say the idea which has been brought in. Definitely that can be done, and only thing is the artificial intelligence totally depends on what you are feeding in. You have to feed in the data, the right data, and then only you can expect the AI to really screen and then get you the answer you're looking for. So it will totally depend that how much information we have about bacteriophages, what we are feeding in, and then you can expect the outcome. And as far as India is concerned, I think we are still in nascent phase. But yes, there are many other institutes which have large number of phages, and they can use AI-based screening first, and then only in those bugs where you really need the phages and where the antibiotics are not working, and then you really know. I mean, you need not be doing everything physically in AI mode, and it will be digital. And like say for just to give an example, AI has screened especially radiologically. They just rule out all your x-rays which are negative, and in seconds, which radiologists will take many hours to rule out, which are negative, and they're only taking positive and maybe which are resembling TB. So you are saving time, money, efforts in using AI-based system. So we can use that AI-based system, but then you need the data to feed it. And the data is the main key. Unless we have data, we cannot use AI.
So ma'am, as you mentioned, so AI has a role in phage therapy.
It has AI role.
But of course, we cannot type in the chat GPT for that give me the right cocktail. We need to give the information to the chat GPT first, then we'll get the answer.
Yes. So you need to feed in the right data to get the correct answer and correct AI applications. But yes, it can be done. I don't know what was the second question. I missed that.
It's about the precision, like in precision oncology, like a precision treatment with phages.
Yes,
again, AI-based.
That is definitely possible. Again, the answer will depend on how good our AI-based system is. Then you can definitely be very precise and giving the right treatment and effective treatment. Yes. But I must appreciate the question.
Yeah, but yes, Dr. Nagwekar has asked a very intelligent,
very intelligent
smart question, role of AI. Everybody is into ChatGPT now. Dr. Sanjay, you mentioned about the CRISPR-Cas. The first time I read about CRISPR-Cas was, there's a book with me, the Code Breaker. I'm sure all of you read the book Code Breaker by the famous writer Walter Isaacson. And that book is on the Nobel Prize winning discovery on CRISPR-Cas by the Nobel Prize winning scientists Jennifer Doudna and Emmanuel Charpentier. And that book is just behind me in my shelf, and a very interesting book. And that book is only about the CRISPR-Cas. And that's, again, as you mentioned, how the bacteria tackle the genes of the bacteriophages and how bacteria utilizing the CRISPR-Cas. Would you please talk a bit more about that CRISPR-Cas technology?
It's essentially is a part of the bacterial immunity. And bacteria have this CRISPR-Cas, which is known as clustered, regularly interspaced short pendomic repeats. They are there in the bacterial genome. So moment phage DNA gets into the cell, and then there are RNAs which will identify if they have the same DNA matching with their RNA, then they will release the proteins. And these proteins will come, they will bind to the DNA and they will cut it like a scissor. So this is how the bacterial cell will be protected against the outside genetic material. But nowadays, people are coming up that this was true earlier, but now this is not true because bacteria, these viruses have got the mechanism by which they can overpower this CRISPR-Cas immunity system also. So, but at the moment, it is only known as a bacterial immunity system, where any genetic material which enters into the cell, the CRISPR-Cas releases the similar kind of the protein which will come, which will cut the DNA and will not allow the DNA to survive. So this is, in broad terms, this is what CRISPR-Cas immunity is. But this has got lots of applications.
Yeah. Can I add something, please?
Oh, yes, ma'am. Yes, sure.
Yeah, so thank you for reading this book. I think it's a wonderful book which you have read, and I will also recommend everyone who is listening to read this book. Now, why I'm saying so? Because the first time CRISPR-Cas was seen was when one of the company who maintains the probiotic strains or lactobacillus strain, they were finding it difficult to maintain their strains for probiotic preparations, which they were using in some of the food articles. And that's the time when they realized that these are the phages which are attacking the bacteria and not letting them survive. And that's where the CRISPR-Cas came into picture. So a lot of research has come into our knowledge, and Jennifer got this Nobel Prize. But basically, it all started with the bacteriophages attacking bacteria, and then, bacterial immunity and the details of bacterial immunity came into picture. So what Abdul said is very, very important. In fact, in the second research day, which as a research dean we conducted at AIIMS, I recommended in the last session to all the students to read this code breaker. And I'm happy that you have brought out that point. It's really eye-opener for all of us. Thank you. I just wanted to add this comment. Thank you.
Thank you, ma'am. Actually, we humans believe we are the masters of the world, but later we learn, no, the bacteria are masters of the world. And now we know, no, it's not the bacteria. The bacteriophages are the masters of the world.
Yes.
Yeah. Our understanding is changing. Our perception of the world is changing. We had 1.5 hours of very interesting discussion, and we learned that the phage therapy is a 100-year-old technique. The Westerners forgot about it. The world forgot about it. But the Eastern Europeans and Russians continued and became masters. And because we don't have antibiotic, the world is now facing a renaissance of phage therapy. And our country is doing, scientists are doing an excellent work in the field of phage therapy. And our funding bodies are giving a very good support, as ICMR is doing very good support. DBT is doing very good support. Of course, as we need to discuss more on the topic, so we still get a better support of it. still more support. And our regulatory bodies know the importance of phage therapy. They are asking our scientists for characterization of bacteriophages and better safety measures for intravenous injection. But they do know the importance of phage therapy very well. We all should discuss more about phage therapy. We clinicians should discuss more about phage therapy. You scientists should come forward and like platforms like this where the audience will be more clinician. We should know about phage therapy, so we can discuss that more with the policymakers, and you will get a better support of, because at the end of the day, it's the patients and the doctors treating know how serious AMR is and how scientists like you need support for technologies like bacteriophages. Thanks a lot for participating. You're all, very busy scientists. We know that. And thanks for spending your time to talk to clinicians and the general audience. And on behalf of AMR Declaration Trust, and again, regarding the trust, and we do have very little funding. So we get unrestricted educational grant from small grants from multiple companies, donors like doctors and other individuals. And again, I take this opportunity to thank the companies like Sanofi, Kalanjan, Converge, Alniche, Fusion, Viatris, and a few other companies for giving us small educational grant so we can conduct such activities without any registration fee. Thanks a lot, and thank you all the scientists. Thanks a lot on behalf of the trust, and we'll conduct more such meetings. We need you back for detailed discussion. This was just an introduction. We couldn't go to any of the details. I know all of you have a lot more scientific details to provide us. We will conduct future meetings. Please attend our future meetings. Thanks a lot.
Thank you.
Thank you very much.
Thank you.
Namaskar, sir.
Namaskar, ji. Namaskar, Rob sir.
Namaste, namaste. Madam, namaskar.
She can't hear you, I suppose.
Thank you, everyone. Actually, I'm
Thank you,
traveling, so there is a lot of net problem, but thank you so much. I really enjoyed today's webinar. Thank you.
Right.
Thank you,
Thank you. Thank you, everyone.